My name is Audrey Belotte, and I've been working in Rolo Lab this summer as a researcher for University of Delaware's Envision program. I'm a rising senior pursuing my degree in pre-veterinary medicine with a minor in animal nutrition. Under Dr. Abasht's lab on wooden breast disease, I was able to isolate DNA from blood samples to study their polymorphisms. Let's meet my mentor.
Wooden breast disease is a muscle disease in chickens and impacts only commercial broilers, which are chickens raised for meat production, and it impacts their breast muscle, and affected chickens has extreme hardness of their muscle, and they are having problem with walking, and the severely affected chickens also shows damage in their muscle fibers. If we look at the histology and see the samples of muscle under microscope, we will see all sorts of changes in the muscle which you cannot see in the normal muscle. For example, sometimes we see severe degeneration of the myofiber. But myofiber are the muscle cells, and we can also see fibrosis if muscle become fibrotic. There are also a problem with necrosis, and we are looking from multiple aspects to see what is the causal factor.
Blood samples from a previous university study on broiler chickens with wooden breast disease were used to begin our experiment. These hatches were divided into 6 different plates, each containing 96 samples of blood. Following the DNA EZ96 protocol, we were able to purify DNA from nucleated animal blood. Our purified DNA products were then sealed into elution microtube plates for storage. We used nanopore technology to measure the DNA concentration of each product and identify an optimal sample. A master mix and primers from the gene TNNI2 were used to amplify the optimal sample H9.
We have done previously genome-wide association studies, which is a method to look at the whole entire genome rather than looking for a specific region. And with this, we want to know if the particular part of the genome is associated with the phenotype. The phenotype in this case is wooden breast disease. So we have done this work. We identified some important region, and the genes that you mentioned, TNNI2, happen to be in one of these important regions, which makes it as interesting genes for further studying.
The master mix, TNNI2 primers, and H9 as our DNA template represent 12 reactions in the PCR machine. They undergo denaturation, annealing, and extension under different temperatures to create new strands of DNA from the primers. Now our amplified samples can be loaded into wells for gel electrophoresis. To prepare the gel, we heat CyberSafe stain with 1% agarose and allow it to cool before pouring. Once this mixture solidifies in the tray, we can remove the comb to reveal our wells. Before loading, we will pour 1x TBE buffer to cover the tray in the electrophoresis tank. A DNA ladder in the first well will serve as a molecular size marker to estimate the size of our DNA fragments. The remaining 6 wells will contain our PCR product and dye. Our gel will run for 40 minutes or until our ladder is fully expanded. Using UV light, we capture an image of the gel to analyze our results against the DNA ladder. Our results show a line in the 5 kilobase pair range, showing the expected molecular weight of our DNA.